thing. So
Why
Carl's
represent
stuff
it
is
as
all
long as
educated. And we've done covariates. But here you've got both
I'm not sure
numeric and covariates.
that.
And so
Perhaps
the
it's
right
even a
thing to
stupid
do,
question.
and then categorical and numerical
I
and
think there is
The right thing to do is to add everything after which you later mention.
maybe. And so and the beauty of it is that that's all for the static connectome. So you can spend a bunch of lifetimes without ever getting to the dynamic connectome, which is what I plan to do.
Spending a lifetime never getting there?
Yes, to the dynamic one. I'm willing to add edge and vertex covariance. But dynamic stochastic processes on these networks, as soon as he added all those feedbacks. Because I was gonna look at that for him, and I said, well, that's gonna be way too hard. I'm too old to do anything that hard. That was brilliant, Albert. Thank you. I have a better guess now for what it's about.
Is this on? Yes.
No, but that was extremely helpful.
That's a broad idea of what the machine learning does.
Yeah.
Unfortunately.
Explode the pin itself into pieces, please. Nothing will make sense if you don't.
There you have it.
Just see if it works.
I'll go to the eye.
Excellent.
How many pieces?
8, 9?
No, he's saying 11, yeah.
Alright, each component is different.
Okay.
Because that gives me a bigger graph, more.
Okay.
Okay, so we're now coming up to our second talk of the day, and we have Marta Zlatic who's going to speak about circuit mechanism and memory-based behavioral choice. She was kind enough to leave the title up so I wouldn't have to remember it. Please.
So this part will be a little bit less circuity, although there'll be a lot of networks in it, and more about what we think these networks are for, which kinds of computations they're doing. So I hope that will be useful as well, and somehow that can inform the broad network analysis as well. And in many ways, it's a continuation of what Albert was talking about. It's putting the mushroom body network into the context of behavior and a larger context of the brain, of the sort of the second order mushroom body network. But what are we really studying? What is the biological problem? All animals have the ability to associate different types of stimuli in the natural world with rewards and punishments. They can learn to predict that one type of a cue reliably predicts the arrival of food, and another type of cue will reliably predict the arrival of a bad thing, punishment. But at the same time, of course, most stimuli that an animal faces aren't a priori completely neutral. So they are rather neutral, but the animal might have an innate like or dislike for something. For example, you can have, you can be innately attracted to many different odors, but then if you eat a food at the same time as you smell that odor that makes you sick, that is poisonous, well, afterwards you'll start avoiding that initially innately very attractive odor. So this means the nervous system must store at the same time these representations, innate representations about innate values of stimuli. And then it must have ways of combining the new learned values of stimuli to drive behavior. And so this is represented here. So when an animal, normally this kinds of neutral stimuli that rather neutral, not fully neutral, that predict punishments or rewards, we call them conditioned stimuli. So if after repeated pairing of a conditioned and stimulus with a reward or a punishment, the animal will acquire a new learned valence for that stimulus. And this learned valence will inform behavioral choice. But as I mentioned, these conditioned stimuli prior to these contingencies in the environment also had some innate valence. So there must be mechanisms in the nervous system that enable these two to interact and the learned valence to override this innate valence. And we don't know how this happens. And in fact, at any given time, the animal is not exposed to a single sensory cue. You're continuously exposed to a whole combination of cues. And these different cues have different innate and learned valences. And somehow the animal must compute a joint integrated valence of all of these cues and then decide which way to go or what to do. And the way in which this integrated valence is computed is also a big open question. And finally, what reinforcement learning theories suggest that what really drives learning is errors between the predicted outcomes of an animal's action based on all of its current knowledge or innate valences, the difference between these predicted outcomes and the actual outcomes. So there must be, whenever an animal chooses to do an action based on all of these combined valences, these are predictions, it also, it drives that, it makes a prediction really about the outcome of its action. Then it performs the action and then it will get a certain amount of reward or punishment based on that action. And then if the reward is exactly as predicted, there is no need to learn anything new, everything was fine. However, if there is a difference in the amount of reward and punishment it received from the prediction and the actual stimulus, well, that's when learning should occur. So, in this computation of comparing the prediction with the actual reward or punishment, it's called the prediction error computation. And it is this prediction error that drives learning in higher order learning and memory center. It drives the changes, it drives the changes between synapse, so the conditioned stimulus will then, based on that, change the value, the strength of its connections to this learned valence pathways. So this is all theory, but in reality we know relatively little about the neural circuits that compute predictions and about so we don't really know how these model corresponds to actual neurons in the nervous system and how these computations are implemented in the nervous system. So in insects, we use the Drosophila as a model system to study this because all insects also, they have a higher order learning and memory center that is in some way for sure implicated in these computations, which is what Albert was talking about. That's the mushroom body. So what the mushroom body, the canyon cells of the mushroom body, they compute the odor identity, and I think Albert probably mentioned that. And then they receive inputs from neurons that receive inputs about different punishments or rewards, and these are dopaminergic input neurons to the mushroom body. So they encode conditioned stimuli through patterns of canyon cell activity, and they receive inputs about punishments or rewards. What we also know, and I think Albert probably mentioned, is that each of these punishment or reward responsive neurons is paired with a specific mushroom body output neuron. And these mushroom body output neurons, they encode the learned valence. But a priori, a particular odor, a particular conditioned stimulus is not associated with any of these learned valence pathways. A priori, the total output of this system is zero. However, what learning will do, based on the activity in these dopaminergic neurons, is change the strengths of connections between the canyon cells and these specific learned valence pathways, such that then a conditioned stimulus will drive a particular learned valence pathway.
A priori, it's zero.
A priori, the total net output of the mushroom body is zero prior to learning. And I'll get to that.
It doesn't know some stuff.
Oh, it has the innate.
Yeah.
That's the, so because a priori there is a parallel pathway.
Oh, it doesn't come through the same path. Yeah.
Which is, this is the learning pathway, and then in the insect brain there is a parallel pathway that encodes innate valences.
I see, so we're not going to change that.
This one doesn't change.
Thank you, thank you, thank you.
This one doesn't change.
Well, it's a bit
This is the one. No, well, it doesn't change with associatively.
No.
And this is the one that changes, and then somehow, and now, this is what we know in the field from a lot of studies from many, many labs. What I just told you is known. But what is not known, it is known that these parallel pathways exist for the innate and the learned valence. What is not known is exactly the way they interact with each other, so that following changes here, you can basically, the nervous system learns to ignore or to do something to this innate valence pathway, so that at the end of the day, it acts based on the integrated valence. What we also don't know, these dopaminergic neurons, well, they receive inputs from punishments and rewards, but as I told you in the reinforcement learning theories, what they actually should be encoding is prediction errors. Therefore, they must be receiving feedback connections from neurons that drive actions and from neurons that compute these predictions. They must be receiving a lot of feedback, so that then feedback can be compared with actual reward and actual punishments. And we don't know the populations of neurons that could encode these predictions and provide this feedback in the insect nervous system. And in fact, we don't know that in any nervous system. So, and I'll go through this quickly. The reason we work in Drosophila larva is because it has a rich behavioral repertoire. It has many, many actions. And you know, we did this beautiful collaboration with Carrie and Joshua a few years ago, where we could, we generated a neuron behavior map of the larval nervous system. We could activate individual neurons optogenetically and just record the entire behavior spectrum, the behaviors that the animals do, and then Carrie and his team developed beautiful methods for classifying behaviors in automated ways, and in just this single context in which the animal was crawling in an empty arena, by activating different neurons, we could get more than 20 different categories of behaviors and behavioral sequences. And of course, if you change the context, you'll get even many more categories. So it has a rich behavioral repertoire, readily amenable to this automated analysis. But at the same time, not only do they have this innate behavioral repertoire, they can modify behavior very well with learning, because they are little insects. They have these mushroom bodies and these networks. So here I'm showing you an example in which we taught the animal to do exactly what I mentioned at the beginning, where initially we exposed the animal to an innately populations of animals, very attractive odors, and you see that we can compute this navigation index, which is a measure of the amount they will crawl towards the odor. So initially these animals innately would crawl towards the odor, but if we pair, so if we have two groups of animals, one group in which we do not pair the odor with a punishment. We give punishment separate, odor separate. And then we have another group of animals in which we every time they get this innately attractive odor, they also get a punishment. They get a painful stimulus. Well, then they exactly invert their preference. They start crawling exactly the opposite way, away from that odor. So we can train them to invert their innate valences. And of course, we have the connectome. I will show you that we can get the connectome and we can reconstruct circuits, and then we can manipulate individual neurons to really determine their roles, their functional roles in these computations. We can manipulate them and see what happens to the computations or to the behavior if we activate or inactivate.
So for the ones that learned to go toward the bad odor or away from the good odor, so you then, your conjecture is that that is in the
mushroom body.
In the mushroom
Yes.
body, and like localized in the mushroom body?
Yes, so I'll get to that.
Okay.
Yes.
Okay.
Exactly to what extent it's localized, we don't know.
Right.
But we do know if we inactivate the mushroom body, this memory they cannot form the memory. What we also know is you can form the memory with specific activation, as I'll tell you, of dopaminergic input neurons to the mushroom body. So we'll get to that. So now, and that's basically, they do this behavior, they can do different forms of learning, and now we want to, first of all, in order to, we have two key questions. How does the mushroom body interact with the innate pathways to compute this integrated valence with innate and learned valence, and how does it compute predictions and prediction errors. So to understand that, we first need to define carefully the functional roles of all of the different dopaminergic input neurons to the mushroom body. We need to build an understanding of the functional organization of this system in the larva. This is already known in the adult, but we need to confirm whether the principles I just told you before hold in the smaller larval nervous system. So each Kagan cell, as Albert mentioned, it forms these two axon branches, and on each axon branch you get these dopaminergic input neurons that tile the branch. So why does it have so many of them? Why can't it just have one dopaminergic neuron that receives punishment signals and another dopaminergic neuron that receives reward signals, and that's it? Why have many different ones? Well, one possibility is that they play different roles. They could encode different punishments or rewards. So there are different types of punishments. There are different types of rewards. So for example, an animal, when it learns that an odor is paired with pain, it could be different to an odor being paired with a specific wasp, or it can be different to an odor being paired to a sound, or different kinds of punishments, different kinds of rewards, different kinds of good taste, and so these neurons could encode the quality of the reward, or they could encode more intensities of the reward. They could encode probabilities of rewards or punishments, as has been suggested in the monkey, for example. So they could encode very complex features of rewards or punishments. So to start figuring out and they could play roles in different memories. So in the adult, it has been shown, and in other insects, that animals can form a short-term memory, a medium-term memory, and a long-term memory. Of course, all of you can do that. And there's different training protocols that evoke short-term or long-term memory. And what has been shown in the adult, that some of the different dopaminergic neurons play different roles in short or long-term memory. So these are all the possibilities. And if we understood very well the individual roles of these neurons, we can then start making better sense of the networks. So to begin to do that, what Claire Eschbach, a really talented postdoc in the lab, what she did is to activate now each of these individual dopaminergic neurons optogenetically and pair it with an odor. Instead of a natural reward or a punishment, she would pair the activity of these neurons with an odor, and then ask, after that, did the animal learn to avoid or to approach that odor? Can these individual dopaminergic neurons substitute for good things for bad things? And if so, do they evoke different types of memory, short-term or long-term? So far, everything I'm telling you is about short-term. We have evidence that some of them are involved in long-term and others in short-term, but this is all short-term protocol. Short-term means you just give the animals 2 or 3 pairings between the activity of that neurons and an odor, and for about 30 minutes, it will recall that that odor was associated with a punishment or a reward. However, if you test it in an hour or 2, it will have forgotten. But if you do the same thing 10 times, then it will remember for much longer, like humans. So all of these neurons, very few pairings of this neuron and this neuron and this neuron, for example, with a reward, it couldn't really activate neither very strong. So, sorry, this is a so-called performance score. And if it is very positive, it means the animal formed an appetitive memory. If it is very negative, it formed an aversive memory. It's a comparison between the preference scores in these paired groups and unpaired groups. So you can see that some of these dopaminergic input neurons evoke is this weird? Well, I think sorry. So some of these, like these ones, evoke very strong appetitive memory, meaning after we pair these neurons with an odor, the animals will very strongly approach it afterwards. Whereas some of these neurons will evoke aversive memory. So So after we pair it with another, the animals will avoid it. And some will evoke neither using this short-term memory protocol.
Looks like in some of those you have multiple neurons that
Yes.
you're activating. Do you think there's some sort of interaction?
Yes,
there, or-
great question. So in fact, what we find is if you activate a single neuron, then the memory is weaker than if you activate combinations of neurons. So this tells us that they aren't exclusively coding quality, that they are acting as a population, and the number of neurons actually encodes probably the intensity and maybe quality. So a single dopaminergic input neuron alone never evokes as strong as memory as their combinations.
Which suggests or tells you that if you had the two connectomes, the difference between the learned one and the non-learned one is
Will be-
complicated.
Not very complicated,
But
but
it's not
it
as
will be.
simple as one.
It will be in more than one compartment.
More than
You
one
you
compartment.
should see the difference in more than one compartment, but depends how you do the training.
Yeah.
If you do the training just with this, you can probably see effects in just that compartment. But if you do the training with multiple, you will see a natural stimulus will affect multiple compartments. And then,
Isn't it?
Yeah. Just a question.
yes.
So how did you choose these particular combinations of neurons to stimulate?
Oh, this set of neurons. These are all of the dopaminergic input neurons to the mushroom body.
Okay, so there are like.
These are, oh, that's from the connectome. So the
Yeah.
connectome tells us all of the dopaminergic input neurons to the mushroom body. So we can systematically test every single
But
one
you haven't
and
tried
all of their
every
combinations.
combination of them.
Sorry?
You haven't tried every combination.
We haven't, but we have tried many, and that's for technical reasons. Because to combine them, you
Yeah.
need to, I won't go into the details, you need a tool that lets you easily combine two neurons. And we don't always have the tool for any combination. We could generate it, but we don't have it right now. And
The optogenetic tool?
no, a galfoline.
Got
I mean,
one.
optogenetic, the effector is always the same.
Right,
crimson.
but
But the selective drivers, so basically we genetically engineer flies so that you can drive only in this neuron or only in that neuron. Now to drive in that combination, you have to combine these two constructs, and that's not always easy. Or you have to be lucky to get a single construct that drives in both, which is what we did in this case. We were just using, we have some constructs that drive in a single neuron and others that drive in the combination. And but this is the summary. When we drive combinations, single neurons to this vertical, this we will call the vertical lobe, this we call the medial lobe. So when we drive combinations of dopaminergic neurons to this vertical lobe, single ones or their combination, we evoke very strong aversive memory. And when we drive combinations of neurons to this medial lobe, we evoke appetitive memory. So basically, the system is very neatly organized. It's got one axon branch for forming bad aversive memories and another axon branch for forming appetitive memories. It's very neat. And so this is what Albert was saying. The same neuron, the same canyon cell is involved in forming many different types of memories because the synaptic changes are local. So you use one neuron for storing many, many memories. And this is how people think cerebellum works as well. And this is why these parallel fiber systems are very economic ways of storing many different types of memories. And now we can go back to our question of, do these different, we know that all of these somehow are involved in forming bad and these good memories, but do they encode different qualities of punishment? Or are they all just encoding the same type of punishment but different intensities maybe, or they just work as a population? And it's a bit of both, like always in biology. They have some, the way we test this is this animal, you can give it different, it has 3, for example, somatosensory punishment types. If we give it a painful stimulus, a pinch, or if we activate the so-called nociceptive neurons, then the animal perform one kind of an escape response. They crawl fast forward. If we activate a different kind of somatosensory interneuron that is actually multisensory, that receives inputs both from noxious and mechanosensory sensory neurons, and really encodes the presence of a predator, for example, a wasp, then the animal performs a different kind of more vigorous escape response. It rolls, it sort of jumps, basically, rolls around its body axis very fast. And if we activate a more milder punishment, such as just a little touch or a vibration, they don't like it, they'll move away, but they do a different kind of an escape response, which is just to turn away. So these are three qualitatively very different punishment types because they evoke three very different escape responses. And if we now look at the responses of the different dopaminergic neurons, we can ask, does one of them respond to one punishment type and the other to another, or do they all respond to all? And the way we do this is we express an indicator, optical indicator of neural activity in these individual neurons, and we optogenetically activate these different sensory neuron types, and we look at the responses of these neurons to these different punishment types. And so you can see here, and color disappeared, but I'll tell you what it is. So these are three different traces. The light is the nociceptive, then this one is the mechanosensory, and this one is the multisensory punishment. And this is the response of this neuron to these three punishment types. This is the response of this neuron to the three punishment types. And this is the response of this neuron to the three punishment types. And you can see that they do a little bit encode punishment quality because they all respond to all punishment types. But this neuron responds more to nociceptive and mechanosensory than to this wasp. This one actually responds more to mechanosensory than to either wasp or noci. And then this one responds more to the wasp-like punishment than to mechanosensory. So they do slightly, they have broad receptive fields, but they do encode a little bit of the punishment quality. And you can also see these reward neurons, the ones that can evoke appetitive memory, well, they don't respond to punishments. They respond to rewards. Now, how do you, so somehow the activation of these neurons together with the Kenyan cells will modify specifically these Kenyan cell to mushroom body output neuron synapses. But now we need to figure out the way in which these mushroom body output neurons encode learned valence. So what do they do? What will mushroom body output neurons do? So they, by encoding a valence, what we really mean by that is that they signify good or bad things, and therefore they will probably drive approach or avoidance behaviors. So now we can ask, if we optogenetically activate individual mushroom body output neurons, will they drive approach or avoidance? And furthermore, as I told you, there are different types of avoidance behaviors. So will these neurons evoke a very specific type of avoidance, or are they still more abstract? Are they still just saying bad or good, and then somewhere downstream, the particular choice of avoidance behavior is made? So we don't know that. So to address this, we can activate now optogenetically individual mushroom body output neurons and look at the behavior of animals and see will they approach or they avoid. So but we want to do this in a high throughput way, not look at one by one. And so how does a larval avoidance look like? How do larvae avoid in a sort of simplest, most general way? So when they're avoiding an odor, which is like a mild, it's similar to a mechanosensory stimulus. There are some innately aversive odors. They're not terribly afraid of them. They just want to get away. So if such an odor comes, if there is a puff of such an odor, or if there's an increase in odor concentration of that type, that will increase the turn rate of the animal. They will just turn away. And when such a bad odor goes away, if there's a drop in its concentration, then the animal knows, oh, I'm going in the good direction. Let's keep crawling, and it will repress turns. So avoidance of an innate odor involves turning to the onset of that odor and not turning and crawling to the offset of that odor. And the attraction towards a good odor is exactly the inverse of that. So if a good odor, if it suddenly feels a good odor, it will repress turns and start crawling. And if a good odor goes down, it will figure, oh, I'm going in the wrong direction, and it will start turning. Yeah. And so now we would expect that if we activate different mushroom body output neurons, that some of them might, the avoidance driving ones, or the negative valence ones, might activate turns to onset of the activity and inhibit turns and activate crawls to offset of their activity. And the attractive ones, the positive valence encoding ones, will do the opposite. They will activate crawls to onset of their activity, and they will activate turns to offset of their activity. But they could even, if they could potentially not only activate turns, but activate rolling behaviors and fast crawls. We don't know. This is if they just encode the sort of mild bad or mild good valence.
And if they outvote the innate system.
Yeah, well, in this case, we are bypassing that's a very good question. If you activate in an innate animal, if you activate all of these together, then their total sum is 0. But if you activate individual ones, that is exactly mimicking the case in which learning has caused a disbalance in this output. And then the assumption is that if there is such an imbalance, if there is strong activity in any of these neurons, they have the ability to outvote the innate system.
Right.
But here also we are not giving any innate stimulus. There's no other stimulus in this animal except optogenetic activation of these neurons. So, but yes, they have to outvote whatever else is. And indeed, we find the expected results. We find that there are some mushroom body output neurons that if we activate them, they trigger, here I'm showing you turning angle as a function of time, and here I'm showing you speed as a function of time. And you can see that some of them will strongly activate turns to onset of their activity and will activate crawls. This is an increase in crawling speed to offset of their activity. So these are the aversive ones. They encode negative valence. And then we have exactly the opposite ones that do exact opposite. They repress turns to onset of their activity. They trigger turns to offset of their activity. So these are the attractive ones. And again, the system is so beautifully, neatly organized. All of these ones to the vertical lobe, they drive these attractive behaviors, and all of the ones to the medial lobe, they drive these aversive behaviors. Now that might be a little bit confusing initially, because I had told you that the dopaminergic input neurons to the vertical lobe evoke aversive memory, but the output neurons drive attraction. The dopaminergic input neurons to the medial lobe evoke appetitive memory, but the output neurons drive avoidance. How can that be? That's illogical. Well, it's not actually, because, and this is exactly the same in the adult Drosophila, that is because the actual change that occurs after learning between the canyon cell and the mushroom body output neuron synapse is depression.
Albert
Good.
told us this story. I didn't
Sorry.
believe it. But if you say it, I believe it.
So basically, forming an appetitive memory means reducing aversion. Forming an aversive memory means reducing attraction. And that's why the system really works as a whole. So a single compartment, it's really what is important is the balance of this entire network. So really this further implies that somehow, somewhere downstream, there are neurons that integrate inputs from all of these learned valence neurons. And they are the ones that drive attraction or aversion, finally, because they need to read out a reduction in attraction and an increase in aversion, etc. So there must be readout neurons from this entire system downstream. So that is what we wanted to find out. That was our original question here. So there must be readout neurons from the entire mushroom body output neuron system, and also there must be neurons that somehow integrate innate and learned valences precisely to allow the learned valence to override the innate valence. And there must be feedback somewhere onto the dopaminergic neurons that will drive these prediction error computations. So can we find that out? And that's where, again, we use electron microscopy reconstruction. So Albert told you that, well, maybe he told you everything, I don't know. But he probably, like this is all of the Kenyan cells, dopaminergic neurons, and projection neurons, and mushroom body output neurons. But then, to identify neurons that integrate the innate and the learned valence, Akira Fushiki, a really talented postdoc in Albert's lab, reconstructed all of the neurons downstream of the mushroom body output neurons. And then many people, this part is published, reconstructed the projection neurons anyway, and then downstream, so this is still in progress, but several very talented students, and including Alex, contributed to this as well, reconstructed the neurons downstream of individual projection neurons in the antennal lobe. And then we could look at the way this innate pathway for innate valence interacts with the pathway for the learned valence. And we could also ask if there are any feedback neurons that feed back onto the dopaminergic neurons. And in fact, we found both of these populations of neurons. We found a large population of convergence neurons and also a large population of feedback neurons that receive inputs from mushroom body output neurons and synapse onto individual dopaminergic input neurons. And so now I'll take you through what can we tell about the system at this stage. So first of all, as Albert mentioned, how is this system read out as a whole? Well, to begin with, there's a lot of interactions between the mushroom body output neurons themselves. And there is a lot of lateral inhibitory interactions between the mushroom body output neurons that encode avoidance and the mushroom body output neurons that encode approach. They cross-inhibit each other, which is exactly what, so basically, if you form an appetitive memory, you will reduce the drive to avoidance neurons. But because the avoidance neurons normally inhibit the attraction neurons, you will thereby also disinhibit attraction. So because of these lateral inhibitory interactions, which is a common feature throughout the nervous system, you will sharpen the readout of your memory. And basically, forming an attractive and appetitive memory involves reducing avoidance and disinhibiting attraction, and forming an aversive memory involves reducing attraction and disinhibiting avoidance. And so that is at this first layer. Then at the second layer, we find this really interesting population of neurons, and some of them are already mushroom body output neurons that we call convergence neurons, that integrate inputs from all of these different pathways of opposite valence. So for example, they receive excitatory inputs from mushroom body output neurons that drive approach. They receive excitatory inputs from the innate pathways that drive approach. And they receive inhibitory inputs from mushroom body output neurons that drive avoidance. And they receive inhibitory inputs from innate pathways that drive avoidance. So they are basically the valence integrator neurons. We call them the convergence neurons. And they themselves are little decision makers, because the way in which this neuron will integrate all of their inputs, it could choose to give more importance to the learned pathway, or more importance to the innate pathway, or more importance to the olfactory pathways, or more importance to the thermal pathways, because it can non-linearly or linearly integrate its inputs. We don't yet know how this neuron integrates its inputs, but these such neurons that drive either approach or avoidance themselves. So they read out the whole system, the learned and the innate system, and then they drive either approach. Oh, I don't know why that happened now, but that's okay. Oops, it crashed.
Microsoft, always helpful.
Probably Russian hackers.
Okay, it's back. So let me show you some evidence for these things that I just said. Okay, so here we are recording activity in this neuron, this type of a neuron, and we can record the activity in a live animal, in a microfluidic device. This is its nervous system. We can do optical calcium imaging in this neuron, and here we are giving it two different innate stimuli, because it receives inputs from multiple innate pathways. There are many good things and many bad things. There's odor, there is temperature, there is light. So here we're just testing in a naive animal, so this particular neuron receives excitatory inputs according to the connectome from odor pathways, attractive odor pathways, and it receives inhibitory inputs according to the connectome from temperature pathways, because cold temperature is something the animals like to avoid. It's an innately aversive cue. And you can see how this neuron has exactly opposite responses. It is, as predicted by the connectome, excited by an innately attractive odor, and it is inhibited by an innately aversive temperature. And now the question is, how will it integrate the combination of these two, linearly or non-linearly? And we don't know that yet, but that's what we're doing at the moment. And then this neuron, as you can see here, also receives inputs from the learned pathways. And we can just functionally confirm that connection. In this particular animal that we tested, so ignore this, the response is the overall, if we activate all of the Kenyan cells, the avoidance drive is stronger than the attractive drive from the learned pathway. So it's actually inhibited by the mushroom body. So it really integrates. This is the neurons we were looking for. Now we can study how they integrate these different types of inputs to make decisions about avoidance or approach. And in fact, there is a whole population of these neurons that Akira Fushiki in the lab identified. And as I say, the way he identified these neurons is by reconstructing everything downstream of the mushroom body out with neurons. And then by picking a few of the very attractive olfactory projection neurons and reconstructing everything downstream of those, and some thermosensory pathways were done by others, and this is how we identify these neurons that receive convergent inputs. And so there are some that if we activate them drive approach, and they basically are inhibited by avoidance pathways and activated by attraction pathways, as you would expect. And then there is the opposite population, which does exactly the opposite. They drive avoidance, and they are activated by innate avoidance driving pathways and learned avoidance driving pathways, and they are inhibited by innate attraction driving pathways and innate and learned attraction driving pathways. And so finally, and this is what we still don't know, but we would like to read out from the analysis of the electron microscopy reconstructions in the network, we would like to identify behavioral choice motifs, or basically the patterns of interactions between these two different populations of neurons. Because if an animal gets a combination of both good and bad cues at once, it will not. Animal decisions are decisive. It will either avoid or it will attract. So somehow there must be a winner-take-all computation that occurs in this network that takes even the slightest net balance in activity of these neurons relative to activity of those neurons, for example, to completely then repress approach and completely drive avoidance. And we have the network and we have the interactions, but we don't, there are multiple kinds of motifs that could implement winner-take-all computations. The simplest one being just reciprocal recurrent inhibition. But it's more complex than that. There is more. So we would love a principled way of reading out the key motifs of interactions between these networks. And then we come to the last question. What about the what about the feedback neurons? Right? The feedback neurons must also be. They must know about the action that the animal has chosen to do. They must somehow know the outcome of this winner-take-all competition. And then they must feed back onto the dopaminergic neuron, because that's the prediction. The prediction is that that action is good. And then they must feed back onto the dopaminergic neuron. So to begin with, let's just look. We did find exactly the right population of neurons using electron microscopy reconstruction. There's a whole population of neurons that receive inputs from specific mushroom body output neurons or specific convergence neurons, and then feed back onto the dopaminergic neurons. And in fact, every single mushroom body output neuron has at least one, if not two or three, feedback neurons that feed back straight to its dopaminergic neuron of its compartment. And then there are some also more complex feed-across interaction that could be playing a role in another kind of a computation. They were a surprise to us, but they're really fun, where there are feed-across neurons that receive inputs from attractive mushroom body output neurons and then synapse onto the compartments involved in forming aversive memories and other way around. And what these could do is, it is a well-known phenomenon in learning literature that often if an animal first learns that something is very good, it is harder for the animal then to learn that that same thing is bad, if you invert the learning rules. And these kind of feed-across neurons, if inhibitory, would exactly do that. They could make it harder for the reward neuron to fire and therefore teach that an odor that was previously paired with punishment now should be paired with a reward. So we have these two types of feedback and feed-across neurons. Now for feedback, of course, we need to now confirm, and this is completely work in progress, that these dopaminergic neurons do really encode prediction errors. Because so far I've shown you their responses in naive animals, and naive animals, they respond to punishments, they respond to rewards. But what is the evidence in vertebrate literature that these dopaminergic neurons encode prediction errors? So this is the examples from pioneering studies by Wolfram Schultz here in Cambridge, who demonstrated prediction error coding in dopaminergic neurons. And so what you can see, and maybe I'm missing one part of the slide, but that doesn't matter, I'll tell you. So in a naive monkey, the dopaminergic neuron nicely responds to the reward, a sugar, whatever it was. But in a trained monkey, this dopaminergic neuron does not respond to this reward anymore. In fact, it starts responding to the conditioned stimulus that predicted the reward. And I'm missing the part here that if you omit the reward, if it doesn't get the reward, the dopaminergic neuron at the time at which it expected it, which would be here, then this dopaminergic neuron will have a negative response. And that's the prediction error. So now we need to give these kinds of experiments to our larvae in the microfluidic device, and this is super preliminary, but we can, and we haven't done it yet in a single animal, but we took just random populations of larvae to see whether the same dopaminergic neuron has different types of responses to the reward in different animals. Do they have naive-like and trained-like responses? Because training the animal in a microfluidic is more complicated. We're doing that now. Now we just took sort of grown-up animals that had different experiences in their lives. And you really do see that the same dopaminergic neuron in one individual, for example, nicely responds, in this case it's not the reward but the punishment, it nicely responds to the punishment, as I showed you before, and it does not respond to odor, and this is like a naive neuron in the monkey, it responds to the unconditioned stimulus, it does not respond to the conditioned stimulus. But here is a different, is the same dopaminergic neuron, but in a different individual, and it behaves like the trained monkey neuron, because it actually does not respond to the same punishment. It instead responds to the odor, to the conditioned stimulus. So it does this,
Why?
basically.
I mean, the same neuron.
The same neuron, because of this, so it's the same in the monkeys, the same neuron. Prior to learning,
Okay, so
it
the-
responds because it changes. The reason is that as a result of learning,
okay.
this synapse changes, and hence this feedback, imagine if this feedback were inhibitory, for example. I mean, it's many inference possible, but because of this feedback, every time learning changes this synapse, it will change the way this neuron responds to a reward as well at the same, it will change the responsiveness of this neuron. Well, because of this synapse, it will change the way it will respond to the conditioned stimulus. And therefore, the way it will respond to a combination of a conditioned stimulus and a reward.
So, to some extent, you just told us where in the connectome the change will be.
Well, that we know.
Yeah.
The change must be here.
Well,
But
but
the
in the connectome, in the overall connectome, it's associated with these neurons.
Exactly.
So this collection of neurons.
Exactly. But as Albert told you, there are also canyon cell 2 dopaminergic neuron connections, and these could potentially explain the occurrence of this response. Or some of the feedback neurons, if some are inhibitory and others are excitatory, some of them could explain one part of the change in the response, and the others could explain the other part of the change in the response. It all depends now on the signs of these connections. But with this network, you could quite easily get these changes. The network can do it very easily. Now we have to, the neurons do change as well, but now we have to figure out whether the neurons that we think are causing these changes are really causing these changes. And if the neurons that are suggested in the network that are involved in these computations are the ones that we have to manipulate them, we have to record their activity to know.
But as soon as you have your hypothesized neurons, you can do that.
Exactly.
Or
So that's
hide
what
the
we're
money
doing.
in the way.
Exactly.
Right? Okay.
Exactly. Whoops. Now this thing decided to go away again, and I don't know why.
Push the button. There's a blanking button in the Marta.
Oh well.
Push again. You have it.
Good. Well, all I want to say is that, to thank the people who did this work. So Kathy Eichler and Lee did all of the reconstructions of the canyon cells, the dopaminergic neurons, and the mushroom body output neurons. Akira Fushiki did all of the reconstructions of neurons downstream of the mushroom body output neurons. And Claire did all of the physiology experiments, and Bruno did the temperature, the recordings in response to thermosensory changes. And of course, our other collaborators that I've been mentioning along the way. And our collaboration with Carrie has been enormously fruitful over the past.
Or annoying, maybe.
Yeah.
Challenging.
Time to speak here, and questions?
Are you going to stay for a few minutes, or are you leaving right now?
Oh, we could
Okay,
we could
well,
stay
hold
for
my
lunch.
questions then.
Yeah.
They have both a larva Olympiad and a adult Olympiad.
What are those?
It's like a genelia major effort, right?
It's like.
Well, not really. So the adult Olympiad was they're quite separate. The adult Olympiad was, it's a silly name. All it means is designing a lot of different assays and then looking for the roles of individual neurons in the different behaviors.
Just a giant effort into the
So it's
neuroscience
not
of-
that giant. So it wasn't such a giant effort, right? In the adult, basically, it was the giant effort was to make all the lines, which is what Jerry
True,
Rubin
yeah.
has done, and then different groups would come. And these assays are very high throughput. That's why we say giant. There's no giant effort.
Okay.
It took Tomoko, for example, for our optogenetic activation screen, it took her 3 months to test every single neuron in one behavioral assay. So it's not so, 3 months to test it. That's in the larva, right? In the adult, they tested 2,000 lines, so it's a bit of a, but we tested 1,000, and you just dump populations of neurons and test them, the response to sound, the response to light. So the effort is then in analyzing the data. And so, which is the adult Olympiad, they're still doing it because they, whereas for the larval Olympiad, it was a few groups from different countries came to Janelia and tested their, for example, there was a group studying innate visual responses, innate temperature responses, innate olfactory responses. So they tested the roles of neurons in those innate behaviors. And then we did like the learned and then all of the somatosensory behaviors.
Here's a question for the two talks. So Albert's got you got your connectome and you got your stereotypical, and except when it isn't, which I still don't understand. And now you have the learn. So let's just as and since we're graph inference, just as a thought experiment. If I observe 2 animals or 10 animals, or I'm only going to get 1, that's purely at the innate.
Yeah.
And then I do the same thing where I've learned one particular thing to go toward the wasp odor or whatever.
That has learned.
Yeah, that has learned. So the connectome is now different.
Sure. Oh, maybe.
Right,
I don't know that.
I know, that's a hypothesis. But if it's different, is the difference stereotypical?
We think so, yes. Based on everything, right? Based on everything we know,
Compartment specific.
the
That's what people
changes
output
will
neurons.
be between the particular canyon cell and a particular mushroom body output neuron.
Well, it won't be the entire alert memory will be on one.
No, particular set of canyon
Yeah.
cells. If you've
Okay.
trained an animal,
So
so
that's
if you, I
what you
can
mean
tell you
by-
exactly how many, about 5. Because if you, no, it is well known that Drosophila canyon cells, when you give them an odor, only 5% respond, right? So there should be 5 canyon cells that will change their synaptic connections in those compartments for those dopaminergic neurons that respond to that punishment.
It's gonna be 1 or 2 or 3 compartments.
So
Yeah,
when you're
and then
looking
it's
at
compartment
it,
specific and just a couple compartments. And
Exactly.
so the conjecture is that memory will show up in the connectome there.
absolutely. It might show up in other places as well.
Because every
But
time you
it
look, you
better
find
show
weird
there,
things.
or if it doesn't show there, the
Right.
memory does not need to be structural. It
Right.
can be purely functional.
I know that
Very
would be,
molecular, yeah.
yeah, right, but, okay, good, thank you.
But-
So let's do that.
Sure, so Ben is,
I know, that's
that's-
Ben's dissertation?
that's why
Yes,
that's Ben's
that's
PhD,
why
but
I'm
that's
here,
why
I know.
his and yours idea to do different memories on different sides of the larva is brilliant, because
Yeah.
then you have,
That's the
the
left, right, so
control.
that's gonna work?
it's gonna
I
work.
hadn't heard that from you yet.
Yeah.
So
That
how
was
is
Bill.
that?
No, so the
It's
left,
gonna work.
right, okay, yeah, go ahead, I don't go.
We
Yes.
have lunch
Oh,
appointment
that's
in town car, so Russell will probably kill me if we don't thank you now and get going to his car. Thank you very much for your talk.